Hello and welcome back to Science of Medicine. I want to talk today about some general concepts in medical physiology that allow us to live. In particular, basic biological structure within our cells, how they communicate with each other, and the processes that allow us to get energy to run those cellular processes. First off, everybody is composed of animal cells. These cells have specialized function, except for certain cells, particularly erythrocytes or red blood cells, platelets, and gametes, either sperm if you're a male or eggs if you're a female. These cells contain a nucleus with a full complement of DNA. DNA, or the chromosomes, half of which you obtain from your mother and half from your father, are made up normally of 46 chromosomes, including X and Y, or X and X if you're female. Each of these cells has a fairly limited lifetime except for neurons, and they arise from stem cells. These stem cells are making new cells because the lifetime is limited. The neurons are exceptions because they take so long to be able to hardwire the control system of your brain and your central nervous system, as well as your peripheral nervous system, that it would be intractable to try to grow them from stem cells and then have them reprogrammed, if you will, to be able to operate the neurological system. The structure and the function of these cells depends upon the use. So cells that make up your bones are going to have different structure and function compared to, let's say, your skin cells. Now, these cells are organized into tissues, and then those tissues comprise organs, and that will provide the overall function to the body. Now, if I look on the cellular level, each of the animal cells in our body are going to be composed of a cell membrane. This cell membrane is going to be a phospholipid bilayer. That is, it's going to have a hydrophobic interior and a hydrophilic exterior, one going to the outside of the cell, the other going to the inside of the cell. And I will have various transmembrane proteins with a glycocalyx, that is a carbohydrate type of material that will allow for cellular communication. Inside the cell, I have cytoplasm, this milieu of viscous components that basically allow for nutrients and other things to be present in my cell. Inside most cells, I'll have a nucleus, and this nucleus will be enveloped by a nuclear membrane, which likewise will have a nuclear pore complex, much like the transmembrane proteins across the cell membrane. This nuclear pore complex will allow for the selective transport of materials into and out of the cell. Now, the nucleus will contain your DNA. That DNA will provide for instructions that will go through that nuclear pore complex to make things, such as proteins. And those proteins are going to be made in many different synthetic domains. There can be proteins made on ribosomes, which are free-floating in the cytosol. They also can be in the endoplasmic reticulum. Endoplasmic reticulum is a sequestered area within your cell that has also a lipid bilayer that will allow for proteins to be synthesized. But those proteins that are synthesized in the endoplasmic reticulum will then be further modified in the Golgi apparatus, where a carbohydrate structure will be placed on it and then modified. And this is especially important for cellular communication and for certain functions. Now, also within the cell, I have mitochondria. The mitochondria, which we'll talk about in a few minutes, will provide a way of generating energy molecules, in particular ATP. Those mitochondria are essential for running the metabolic processes of our cells. I have certain kinds of containers. Lysosomes, peroxisomes, and vesicles are examples of those. Those could be storage containers, such as vesicles, which contain, let's say, neurotransmitters. They could be degradation encapsulated containers, such as peroxisomes, which will help to degrade things. Or I could store things that then get dumped into the exterior of the cell, which can degrade things. I I have to have a way of moving things back and forth in my cell, or even moving my cell properly. And those cytoskeletal elements help with this. I have microtubules, actin, and intermediate filaments as examples of those. Microtubules are put together and taken apart to be able to allow me to transport things. For example, I have microtubules which are present in the axons of neurons that allow for neurotransmitters to be transported all the way down to the end of the axon. I have actin and intermediate filaments which also provide structural integrity to my cells as well as motility. I have to have a way of communicating with different cells. So if I'm a cell and I'm present in a certain environment, I'll have adjacent cells. I'll have cells that are further away in some other tissue. And there are different general ways of communication. So if a communication occurs via secreted molecules, I would have different possibilities. If I secrete a molecule that binds with the same cell that is secreting it, I call that an autocrine form of communication. You might think, why would this be essential? Sometimes you want to have positive feedback mechanisms where a cell will self-stimulate itself to do more of a function, a particular function. I have the capability of secreting a cell, and then a nearby cell will have that molecule bind with it that will cause some change. That's a paracrine type of cellular communication. Some cells, specialized cells called endocrine cells, will secrete a molecule into the bloodstream, which will then travel to another cell and bind and then affect whatever it's supposed to do. And that's an endocrine form of communication. And then another specialized form of communication is synaptic, where I have a neuron will release a molecule or molecules that will cross a small gap called a synapse and bind with some cell surface receptors on an adjacent cell. Could be another neuron, could be a muscle, and then that will have some further effect. And we'll talk about those different types of cellular communication throughout this Science of Medicine course. Now, different cells respond to the same molecule differently. So I can have some cells that have acetylcholine. When that's released, it can lead to the contraction of skeletal muscle, but it decreases the force of contraction of cardiac muscle. So when you think about the molecules that are used for these different types of cellular communication, don't think this molecule does this. That's not in fact the case in the body. And remember that those effects depend upon binding to something on the surface of another cell or on the same cell, and I can upregulate or downregulate those molecules. Now, there are lots of different signaling pathways. I'm going to talk about several major pathways. But the more we investigate, the more we find that there are different pathways. Small molecules, things like nitric oxide, have been found to be pathways for cellular signaling. But I'm going to talk about 4 major types. 3 of them are related to a molecule binding to an exterior cell surface receptor. And one of them is an intracellular surface receptor. So the 3 that are exterior cellular surface receptors are ion channel, enzyme link receptor, and G protein receptor. Now, in an ion channel receptor, a molecule will bind to a receptor on the cell surface, exterior cell surface, and that leads to a conformational change that transiently opens up some type of channel, like an ion channel for potassium or for sodium. And that ion, if there is a gradient, a difference in the amount of ion on the inside and the outside of the cell, that ion will flow spontaneously into or out of the cell, and that ionic change will lead to an effect, such as contraction of the muscle. In this case, the ion crossing the membrane is the messenger. An enzyme link receptor will have a molecule bind to a receptor on the cell exterior portion, but it activates an intracellular enzyme. And there are several intracellular enzymes that are used. It could be cyclic guanine monophosphate, or GMP, tyrosine kinase, or RAS proteins. And those three secondary messengers will then cause the effect of whatever's going to happen based on the binding of that molecule. In a similar way, there are transmembrane proteins called G proteins. And when a molecule binds to a receptor on a G protein, it actually allows for the binding of another protein on the intracellular side of the G protein, and that leads to the activity and the secondary effect. So the secondary messenger, such as cyclic AMP, or calcium, or inositol triphosphate, ITP, those three effectors or messengers are what are causing the downstream effects. So these are linked to G protein receptors as opposed to the secondary messengers that are linked to enzyme-linked receptors. It's very important that you understand and keep these separated because we have a lot of medications that are tied to interfering with these different protein pathways. Now, intracellularly, some molecules, such as steroid hormones, as well as hydrophobic molecules, nitric oxide, vitamin D, can move through the cell membrane and go directly and bind to DNA sequences, for example, for regulating genes. And these affect intracellular processes directly. There's no mediation through some extracellular binding domain. Now, sometimes in these processes, we have phosphate groups that are added to a protein. Sometimes it activates it, sometimes it deactivates it. In general, if you add a phosphate group, it tends to activate it, but that's not always the case. However, proteins that add a phosphate group to some other protein are called kinases. Those are enzymes that add a phosphate group to a protein. Phosphatases are a set of enzymes that remove a phosphate group. Many times this deactivates it, although that's not always the case. And sometimes calcium will regulate these effects. For example, calcium can bind to calmodulin, and we'll talk about that when we talk about smooth muscle. Now, in order to carry out these functions within my body, I have to have energy. And I don't have little batteries. What I have are energy molecules. Molecules that when you cleave a bond, typically that's a high energy phosphate bond, it will release energy that can be coupled to another process that is not spontaneous. So I do that with things like ATP or adenosine triphosphate. I can cleave a phosphate bond and give adenosine diphosphate in a phosphate group, but more importantly, energy, or guanine triphosphate. And that's a way that I can actually have energy. I can have energy that is bound to certain molecules that act as electron stores, such as NADH or FADH2. And I can also have electrochemical gradients that arise from ions that could transfer across a membrane. In particular, protein gradients that are made from things that come out of the Krebs cycle will create a proton gradient in the mitochondria, and that's used to synthesize ATP. Now, you don't eat ATP or GTP or NADH or FADH2 or proton gradients. You don't eat those things. What we eat are foods that are comprised of 4 different categories of things: carbohydrates, proteins, fats, and everything else, which we call roughage or fiber or whatever other undigestible material term you want to actually use. Now, the undigestible material, the fiber passes through you and goes out in your fecal matter. But the carbohydrates, proteins, and fats can be broken down and absorbed and used as feedstock for being able to generate energy or building blocks that are necessary in your body. So I want to talk a little bit about each of those. So in the case of carbohydrates actually will break down and produce glucose or some sugar. Now, fortunately, in your body, you have lots of metabolic pathways. So if you have a 6-membered sugar like glucose, but maybe it's bound to some 5-membered sugar, then you can transfer back and forth between 5 and 6 membered sugars without any trouble. There are different pathways that you can use. Glucose ultimately is broken down via glycolysis to pyruvate. That means you're taking a 6-carbon moiety and breaking it down into 2 pyruvates or 3 carbon moieties. Under aerobic conditions, that is when you have oxygen present, which is most of the time, the pyruvate then gets oxidized to acetyl-CoA, and that acetyl-CoA is fed into the Krebs cycle. The Krebs cycle, carboxylic acid cycle, that acetyl-CoA metabolizes to carbon dioxide and water. So ultimately, the carbohydrates, if they are metabolized completely, will actually break down into carbon dioxide and water, which you can release by breathing out or by excretion in the case of water. Now, in this Krebs cycle, I don't generate ATP directly. I do generate things that can be used to make ATP. And if you do some estimations of what that would be, you end up accounting for about 36 ATP molecules for a molecule of glucose. Sometimes my body has to operate temporarily under anaerobic conditions, or sometimes, like in the case of erythrocytes, I don't have the machinery to do the Krebs cycle, and when I operate under anaerobic conditions, I use the Ebden-Meyerhof pathway, where I'm just basically taking the pyruvate and making it into lactate, and I just get the 2 molecules of ATP for glucose that I get from this glycolysis of breaking down glucose into pyruvate. So not a lot of energy, but it gives me some. Sometimes a fatty acid is made up of an odd number of carbons, and in this case, I'll be left with, instead of an acetate, I'll have a C3 or a propionate. And there's a specialized pathway that converts that into succinate, which is one of the substrates that's used in the Krebs cycle. Proteins, on the other hand, give me amino acids that are the building blocks for proteins within my body. Under normal conditions, I'm not trying to use proteins to generate energy. I'm using them for their structural components of the amino acids. And there are about 20 different pathways that lead to the 20 different amino acids, and all of those will form products that will enter the Krebs cycle. However, when I degrade or catabolize proteins, I have a problem. These proteins contain amino acids which contain nitrogen, and the degradation product is ammonia. But ammonia is toxic. So I have to have a way of converting that ammonia into something that is non-toxic, and the molecule that it's converted into is called urea. And that urea cycle is what actually will allow for that conversion. That's done in the liver. So proteins are generally broken down not so much for the energy, but more for the component amino acids that they have. But their degradation leads to this toxic byproduct, so we have to go through this additional process in the urea cycle to convert it to something which is less toxic and can be excreted. Let's do some applications. So fluoroacetate is a poison that is derived from many kinds of plants. If you take this in, if you ingest it, it binds to acetyl-CoA, but the subsequent reactions are rendered the acetyl-CoA unreactive in the body. That is, it stops acetyl-CoA from reacting further, which is following is likely to occur if you are poisoned by this fluoroacetate. Do you generate excess beta-hydroxybutyrate? Do you have ammonia that builds up in the blood from protein catabolism? Do you have decreased energy generated via the Krebs cycle? Or do you inhibit glucose conversion to pyruvate? So I hope that you noticed that you will have decreased energy via the Krebs cycle. Remember, acetyl-CoA is fed into the Krebs cycle and will allow for ATP to be generated. These other processes are not directly affected. How about this? Gleevec is a tyrosine kinase inhibitor. That's a drug that will bind to tyrosine kinase and makes it so that it doesn't work. And that's used in the cancer treatment for chronic myeloid leukemia, CML. It's a type of blood cancer. Which of the following signaling pathways is most likely involved? Steroid hormone, ion channel, G protein, or enzyme link receptor? So I hope you recognize that it is enzyme link receptor because it's a tyrosine kinase inhibitor. Remember, tyrosine kinase is a cellular signaling pathway of enzyme link receptors. So I'll come back and talk to you about some additional information about basic kinds of cellular processes, in particular related to fluid compartments and homeostasis in just a few moments. I'll see you then.